Hemoglobin is composed of 4 subunits. Additionally, there are heme groups which absorb oxygen. Hemoglobin is composed of 2 main subunits, alpha and beta globin. Altogether, the functions which hemoglobin serves is allowing molecules like oxygen to bind to the red blood cells and to be transported where it is needed in the body. Scientifically, hemoglobin is characterized as a globular protein. As for the action within the body, hemoglobin acts as a transport protein carrying oxygen across the body. Almost all advanced organisms which have blood have hemoglobin and its derivatives within the red blood cells. The hemoglobin itself is located throughout the red blood cell. There are a few organisms which do not contain hemoglobin, including simpler life like single-celled organisms, as well as a very few set of actual vertebrate animals which simply evolved to meet very specific conditions where hemoglobin was not needed. An example of this is the sea cucumber, which rather than using hemoglobin uses something called hemocyanin. The mutation causes the protein to become less active. This is due to a region which is originally hydrophilic being replaced by a hydrophobic molecule. As a result, this causes the hemoglobins to clump together, which warps the shape of the red blood cell, making it less effective at transporting oxygen as intended. The mutation occurs within the beta globin protein within the hemoglobin subunit. The highlighted regions on the screen are the beta globin subunit proteins, the location where the mutation occurs. This change itself does not physically change each individual hemoglobin molecule too much. However, the change from being hydrophilic to becoming hydrophobic can cause the hemoglobin molecules to clump, therefore distorting the function and shape of the actual whole red blood cell. In a normal hemoglobin protein, under normal conditions, a glutamic acid molecule is located in the area. However, after the mutation occurs, the glutamic acid is then replaced by a valine. Glutamic acid is one of the only amino acids which carries a negative charge. Along with that, it's also a highly polar molecule. As a result, glutamic acid has a high affinity to water and other polar molecules and can create hydrogen bonds. Additionally, ionic bonds are a possibility for glutamic acid. This means that when the mutation occurs and the glutamic acid is replaced by the valine, there are big changes as the valine is hydrophobic and nonpolar. On the genetic level, the change which occurs happens in a chain of genetic material which is originally GAG, which becomes replaced by the sequence GTG. This is responsible for the change in the replacement of glutamic acid by the valine amino acid. While the model which I've created is still 3D, it is a simplified version which focuses more on the two-way symmetry of the protein. The true shape of the hemoglobin protein is across the 3D lattice with many small twists and turns, and it would have been too difficult to create this in real life as a model, which is completely accurate. However, with that being said, my model is still accurate as it includes all of the most important main structures like both alpha and beta globin subunits, as well as the heme and oxygen molecules which they are able to hold.